So this is our today's topic: protein labeling and staining using fluorescent dyes and new opportunities in the electrophoresis based in protein analysis. So what is the big problem when you are talking about sensitivity and sample amount? So what you see here is the normal range of abundances of different proteins you can have in your sample. This was reported by Anderson in 2002, and what you see additionally is that the abundance is different depending on where your protein is come from. So this comes then to the demands that you have on your protein detection chemistry. So first of all, what you want is a high sensitivity. And of course, it should be quantitative, with a wide linearity. What this means, I will explain later on in another slide. Again, a wide dynamic range would be great. And if you talk about the upscaling, upstreaming of your method, it perhaps should also be compatible with mass spectrometry. This is quite new in the last years, with the reports of stains and dyes, and they are toxic, so it should be no toxicity inside your chemistry. This is new from the last years, and it is environmental friendly. So these are a lot of demands from the side of the lab and the side of your wallet. It should be fit into your budget. So the more expensive a chemistry is, the less you would do with it. So a nice budget with a low price is always a good point. So how are proteins in gels detected? There are different possibilities. You could, for example, pre-label your proteins. So this is mainly done with radioisotopes like 35S. You can use fluorescence, the topic of today. There are a lot of different fluorophores available on the market. The most important ones are for sure the DIGE stains, Sci 235. There are competitors, and they call these stains Refraction 2D. And of course, SERVA has also some fluorophores in our portfolio. We called them SERVA Lightning, Sci 235, and HPE Lightning Red. But you can also stain your proteins, for example, with organic dyes. That would be Comasi silver. And you also have fluorescent dyes. So this is a difference between the DIGEs and the lightning reds and so on. You can dye them directly with fluorescent. This would be, for example, our SERVA Purple. And the reverse staining is also possible. So if you do not want to stain your protein at all, you could stain the gel background. This is done via an imidazole zinc staining called SERVA Snow in our case. We have this in our portfolio as well. And the last one would be blotting. There you have different possibilities, for example, immuno or affinity detection on a blot. And here you have all these detection methods as a table where you can see the sensitivity limit, the quantification, and the linear dynamic range. So what does that mean? For example, you have the Comasi blue staining. There you can detect proteins with yeah, the mass of 20 to 5 nanograms, depending on what kind of Comassi you use. The 20 nanograms is the normal Comassi staining. The 5 nanograms is the colloidal staining that is available. The quantification is okay. It's not the best, but you can do it if you like. But you have a small linear dynamic range, and this linear dynamic range is also importable for quantification. If you are out of the range where it is linear, you get false results. So the bigger the linear dynamic range, the better it is for different, talk about proteins, loads of proteins. The negative staining, you have a sensitivity, so this is zinc imidazole one of 15 nanograms, but you cannot quantify it. Why not? Because you stain the background. So you do not have any clue how much protein you have per band. And therefore, you also do not have a linear dynamic range. So the most popular one, I think, is the silver staining. This is the workhorse in most labs. There you have a sensitivity limit of 200 picograms, so much less than you have with Comassi, but it's not quantifiable. Why not? Well, if you have done silver staining before, you know that you have to stop the staining. So, and this is the big problem. You never know when to stop, even if you use a time point setting. Let's say I stop after 2 minutes or something like that. You can never be sure that between the different gels, you have the same stopping. And if you forget to stop, you get a black gel. So, and this is also the reason why you don't have a linear dynamic range. It is not mathematically calculatable, the dynamic range, because of this stop reaction. With the fluorescent staining, you have a nearly good sensitivity like in silver, so 400 picograms around that, but you have a huge quantification, better than Comassi, and the linear dynamic range of 10 to the 4 is also better than with Comassi, and this gets topped by fluorescent labeling. So there you have a few picograms and an even more quantification value with even more linear dynamic range. Of course, all the radio and isotope labelings you have, so like an X-ray film, the phosphor imager, or stable isotope labeling is also a very good application compared to the fluorescent labeling. But as you can imagine, everything that has to do with X-ray and stuff is not good for your health if you do it wrong. So we say that fluorescent labeling is perhaps the best and the safest one that you can use in your lab. So how is fluorescent staining made? There is a labeling, and you have a fluorophore, like shown on the left, and you just enter energy in, let's say, as light. So light goes in as energy, and it raises the energy level of your fluorophore to a higher level. This is called excitation. And with a little loss of energy, it emits another light. So, but this is not the same one, it's a different one. So it also depends on point 2 here, the loss of energy. So it could be that you put in some color here that has a defined wavelength, but with the loss of energy, the wavelength gets mainly longer or wider, and you change the color that is emitted. So this is what you see in this graph. That's the frequency of the event and the wavelength of light that is on the one hand excited. This is the blue line here, and emitted, it is the red line here. An also important factor is always the difference between the highest points of the excitation and emission curve, because if you use a filter to get rid of the excitation light and only image your emission, this gap or the width of this gap is quite important, that you can easily get rid of the excitation light. And this is the benefits, just as summarization, of fluorescent detection. So you have this high sensitivity, the wide dynamic and linear range. You can multiplex it. We come to this on another PowerPoint later. It's a very fast procedure, and these are the general protein stains we find in most labs. So these are called SciPro. For example, you have different colors like orange, red, rose, ruby. They call it flamingo, krypton, and the SERVA Purple, it was called before, lava purple, and deep purple would be the protein stains. And we also have a very good one that is called Fluo-Y. And these are the labels, the fluorescent labels that are quite popular on this market of the fluorophores. And these are, of course, the SciDyes, Sci 2, 3, and 5, that are labeling lysines and cysteines, refraction to D, or, as I told you before, our SERVA Lightning Red and the Lightning Sci Dyes 2, 3, and 5. This is our SERVA protein stain Fluo-Y. It has a sensitivity nearly to 1 nanogram per band. It's linear quantification over 3 orders of magnitude. It's directly detected by an UV table or light source. So this could be of advantage if you already have an imager in the lab that is used for, let's say, DNA, RNA detection. For example, with ethidium bromide, you can use this staining, and it is mass spec compatible, and the dye is, that is a big advantage, removable from the gel by washing with water. And here you see just a small example, a dilution series of BSA, where you can see that you have a 5 microliter volume per lane of this dilution series, with this side 2.5 micrograms to down. Perhaps you can see it on your screen slightly, 1 nanogram. Then this is our SERVA Purple. This can be very useful with 2D page. We have another webinar online where Christian Moritz also used it for the detection. With the detection, and the sensitivity here is as low as 50 picograms. You have a linear quantification over 4 orders. It's compatible with mass spec, Western blotting, and all your upstream you have. It's very environmental friendly and easy to use. And as a sample, you have a 2D page gel of an E.coli extract, where the first dimension was made on an IPG blue strip and the second dimension on an HPE large format gel with 12.5. Again here, a SERVA Purple staining with an pH 5 to 8 gradient. This is 100 micrograms of E.coli in a 2D gel flatbed large. Also very nice, you can see all the round spots. And another possibility where you can apply SERVA Purple to this would be a normal SDS page of E.coli and HeLa cell samples, so you can also use it with a normal SDS page, not only for the 2D electrophoresis. Quite new fluorescent labeling, so we invented this mid of last year around that, is our SERVA Lightning Red. You can have it as 1D SDS page solution. It is a very simple and stable labeling reaction that binds to epsilon amino groups of lysine. It has a high sensitivity of 100 picograms per band, no bleaching. The dye is stable for weeks, and then I can say you it's weeks. We still have gels in the fridge that are working quite well, and with a very, very good competitive price. And this is how it works. Normally you have Comasi blue silver staining or even SERVA purple. You do your electrophoresis, 1D or 2D, depending on your application. And you see, for example, the Comasi blue after fixing and staining and destaining, you get images around, yes, nearly 2.5 to 3 hours. Even worse, it's silver staining. Then you fix, you wash, you sensitize, and so on until you develop and stop. You get an image after 4 hours. SERVA Purple is a little bit better because you just have to stain, wash, and acidify, but nonetheless, it's 2.5 hours before you get your first image. So there is a difference of Lightning Red. The staining is done in advance. So you label your proteins before your electrophoresis, and this is around 30 minutes. You do your electrophoresis and you go directly to the imaging system you have. Afterwards, you can decide to fix it so that the gel is stable for a longer time. And the label procedure is quite easy. So you reconstitute your dye in DMF that is delivered with the aliquots. You mix well, spin down every time. Then you mix the sample with an equal volume of your sample buffer. The recombinant protein concentration is around 1 to 10 milligrams per mil, and you heat the sample for 5 minutes at 95 degrees. Prior to the labeling, before you put it to your sample, you mix the dye 1 to 50 with the reducing sample buffer, and then you go. You pipe it then 10 microliter of labeling solution to your vial with around 5 micrograms of proteins. Here's an example how to do it with 5 microliter sample of 1 milligram per milliliter. You mix gently, and then you place the vial at room temperature for 30 minutes. It is not the worst thing to put it in the dark, or in dark sample tubes or whatever. You mix gently and then apply the labeled protein sample on your SDS page gel. For example, then in our example above, 5 microliters per well. The SERVA Lightning Red wavelength has an excitation of around 530 nanometers, so it can be excited with a blue light, and an emission filter of 610 around that, with a narrow band of 30 nanometers. And this is a comparison we have done before. So you have on the upper left side the Comasi blue staining, right beside it a silver staining, then on the bottom left the SERVA purple, and on the bottom right side the pre-labeling with the SERVA Lightning Reds. Always the same sample, same line, and so on. And what we have just a test mixture of it with 5 micrograms to 1.6 nanograms dilution series, and an E.coli lysate with 25 micrograms to 125 nanograms dilution series. And what you can see is what I had explained before. While Comessi Blue is quantifiable, as I told you, the sensitivity is, let's say, here the second dilution series, and here the third one with silver staining. This is a factor more, but not quantifiable. On SERVA Purple and Lightning Red, you have this good linear dynamic range, and even with SERVA Purple, you have a better sensitivity, let's say line 5 around that, than silver staining, and even better, and a very nice picture we have here, the pre-labeling with the SERVA Lightning Red. Another example here we have BSA in nanograms from 5,000 to 0.9, and as you can see, the Lightning Red can go down to 1 to 2 or 1 nanograms. Another example, it's an E.coli lysate. Of course, you have much more bands, so you cannot go down to 1 nanogram, but you reach, let's say, 30 nanograms with E.coli lysate. And the last example of this, it's a mini 2D electrophoresis. We had loaded in IPG strips with 10 micrograms total protein in the first dimension. In the second dimension, we used our prime gels, and you see that you can detect very well mini 2D electrophoresis with our Lightning Red. Here you have an example of urine samples. This is a silver stain versus the Lightning Red. So on the left side you have urines, and again our test mixture, and here Lightning Red labeled samples. And if you work in diagnosis and have urine samples, you get even better and more reproducible results. As you perhaps can see here in line 5, you get a signal where you don't have one at silver staining, so it improves your application even more. And this is an example of a pre-labeling with Lightning Red with SERVA HPE Lightning Red. This is a little bit different. This is a different pH gradient. So this is the second product we have in our Lightning Red portfolio, because when using or when doing 2D electrophoresis, you have to be aware of the pH that you use. And after the labeling of these proteins, so they were labeled in advance as well, so 30 minutes before you put it on your IPG strips, and even it stays stable when doing a focusing of the IPG strip. You go on a second dimension with our HPE gels, and this was then scanned with a Typhoon. But the Typhoon has a big problem. It is a very expensive unit, so mainly you go to a core facility or to another lab, or you have enough money to buy one. So this is a laser scanner. Just to summarize here, again, the Lightning Red advantages of our HPE Lightning Red. You have the direct detection without destaining, staining, washing steps. Go out of your electrophoresis, put it on your imager or scanner, and there you go. You can process it further. For example, you can Western blot it, and you can also detect it on the Western blot afterwards. You have a very high sensitivity. You can go down to, let's say, 100 picograms protein per spot in 2D electrophoresis. You have a wide dynamic and linear range up to 10 to the 5. You have no overstaining effects like the MR shift or else. The labeling is quite unaffected by amphalites and reductants, but the pH should be above 8. This is what I explained before. And of course, not only Western blotting, but it's also fully mass spec compatible. Yeah, contact us for a free sample if you want it. Of course, with all the other products we've shown you before, the stains, the gels, just contact us and get your free sample. Now I want to present another labeling we invented just some months ago. But before I do this, I want to explain a little bit about 2D DIGE, so the difference gel electrophoresis system. The first paper here was reported in 1997 in the journal Electrophoresis. And what is this DIGE? You have your samples, and this is mainly a control sample and a treated sample, and you can pool them to have an eternal standard. So 1 to 1, you pool it. After that, you label them with 3 different fluorophores. In this case, they were called Sci 3, 5, and 2. One is red, one is blue, one is yellowish. And then when you label these proteins, you pool them again. Why do you do that? When you pool them, you get rid of all disturbances and false results because you level them between the different samples. Also, perhaps you have some errors in pipetting and so on and so on. You can get rid of this. Then you put this mixture on your IPG strip where you perform your 1D electrophoresis, so isoelectric focusing, you can say. And afterwards, you put this IPG strip on a normal SDS gel and let it run, and you get your 2D gel. Afterwards, you scan it with the different wavelengths of the red, the blue, and the yellow. And what you get is single images of your mix sample where you have some green spots, some red spots, and some blue spots, each in a single data set. And then you do a false color representation. As example, we have here mouse liver proteins. This would be such a picture with the Sci 3, so it's getting green. You have Sci 5 as red and Sci 2 as blue. And afterwards, you do an overlay of these 3 pictures, and you get such a appearance here, where white would be spots where all the 3 colors are present. So these would be the spots you are not so much interested in because they are equal in your sample, your untreated sample and your treated sample. But very important would be the red and the green colored ones. That's meaning that a green spot would be overexpressed in, let's say, your untreated sample, and after treatment, the spot would be overexpressed here as a red spot, and you can therefore improve a lot your protein electrophoresis, and you get much more information than a single SDS page, of course. There are 2 ways of labeling. You have a minimum labeling where you only label the amino group of the lysine, and it's a charge and size match, so you have no multiple label possibilities, and you label 3% of the total amount of all your proteins. And then you have a saturation labeling where you label all of your cysteines of all the proteins in, so 100%, let's say, of the proteins in your samples. there you have a neutral size match charge, and multiple labels are possible, and it is extreme sensitive. But to, let's say, 80 to 90% in the labs, they only do minimum labeling because they can also add the side 2 and get even one factor more of information out of it. And this is just the chemistry, just to show you how minimal labeling works. So you have your protein and the amino residues. You have an NHS ester reactive group. This would be your SciDye, and with pH around 8.5, 0 degrees for 30 minutes, you get this esterization of the dye to the protein. So just to summarize again, only 3% of the amount of all proteins are labeled. Your multi-labels are statistically below the LOD. The sensitivity is still comparable to silver staining. You can downstream, you improve your downstream, like the mask spec blotting, and with 97% of the non-labeled proteins. And the 2D image is the same like from the non-labeled proteins. The labeling of all the lysines would create a hydrophobic, very hydrophobic proteins. And just if you are not sure if you understand that, you can of course ask us afterwards. Or this book is a very good one. It's made by Rainer Kramer and Rainer Westermeier, a very dear colleague of us that is retired now, but still we are in contact. So if you get an issue of this book, just grab it and read much more about this DIGE protocols. But I want to go back to what we can offer you and what we can show you more. The SciDyes are very, very expensive. So if you are low in budget, or if you want to try something cheaper that is in the performance the same than the SciDyes, we present our Lightning Sci Dyes. We just call them like that. We also have them as 2, 3, and 5, and it's a very, very good budget solution for you. So the chemistry about that is the same that in the original SciDyes. They are for minimal labeling. In 2D electrophoresis, you have the size and charge match fluorescent dyes, of course, for mass spec. They are compatible with all images that are also used for the Sci 235 dyes, for example, a typhoon scanner or whatever you have. The gels that are stained with Lightning Red are ready for the mass spec analysis. The labels, the labeling is labels all the available lysines and would make this hydrophobic proteins. 3% of the amount are labeled. It's all the same than in the SciDyes, and therefore it's also the same protocol as for the SciDyes, as both substances are the same. And if you come back to us, we tell you the price if you are interested, and you will see that the budget is absolutely compatible. Here are pictures we took of our dyes. Oh, I see there's a typing error. Of course, this should not be SciDye, it should be SciDye. So here is the blue picture, the green, the red. It's done with a black and white picture, so you don't have colors, but we put them into false colors afterwards and made the overlay. And here is a second one, second test we did. No, not we did it. Dr. Olaf Kniemeyer from Jena did it. He's using our SciDyes a lot, and he made an E.coli lysate and labeled it with Sci 2, 3, and 5 in the first dimension on a blue strip, a second dimension on our SERVA HPE large gels. And here you see the three pictures of the single colors and see that they are really good and see a lot of spots. You have a lot to do afterwards. So, but we said, well, if you have a good budget solution for you, then we should offer you as well a good possibility to detect those fluorescent dyes. And there comes the SERVA Bluelmager in our game, because as I told you already, the scanners that are mainly used are that expensive over 10,000, 20,000 of euro and even more. So we were searching for a good alternative, and this is our Bluelmager. He looks like that, and it has an CCD camera inside with op to 3.60D. You can set up your exposure time in 2 Mbit pictures. It is equipped with an RGB table and a white light table. It makes autofocus and has a filter turret wheel where you can enter different filters if you like. So it comes with a 595 filter for our SERVA Lightning Red, but can also optionally equipped with 2 more filters so that you can make pictures of the SciDyes or SciDyes. The image storage is quite easy via a USB port up here, or even better, you can have a Wi-Fi option that is installed as well. So via wireless LAN, you can take pictures and put them directly on your tablet, iPhone, smartphone, whatever you have. Of course, it is C certificated. And here we did a comparison, the very expensive Typhoon scanner and our Bluelmager, and you see that you nearly see any difference between the intensivity of the single spots here. It comes with a white light table as well for colorimetric stains. Here you have such a colorimetric stain. This is an isoelectric focusing gel stained with SERVA Violet. SERVA Violet. And here is a normal 1D page, so a normal SDS page that was stained with Lightning Red on our imager.